Did you know that the cell membrane of your body cells contains specific markers which are more like ID cards? And this is how cells recognize each other. That is why our immune cells do not normally attack our body cells, and they only attack the foreign cells that get inside our body. In a previous lesson, we discussed the cell theory, and we know that the cell theory was originated in the 17th century due to the invention of microscopes. But it was during the 19th century when a complete cell membrane theory was developed. This means that for around 200 years, scientists were still not sure about the structure of the cell membrane. By that time, and based on a series of experiments conducted by many scientists, it was confirmed that the cell membrane is composed of proteins and two lipid layers, a lipid bilayer. However, the role of proteins was not yet confirmed. In 1935, Hugh, Davson, and James Danielli proposed the first model that attempted to describe the position of proteins within the lipid bilayer found in membranes. Their model was called the protein sandwich model. Let me explain why it was called a sandwich. Davson and Danielli proposed that there is a layer of proteins adjacent to the phospholipid bilayer on each side of the membrane. As a result, the protein layer is like the bread in a sandwich, and the lipid bilayer in the middle is like the inside of a sandwich. Electron micrographs showed a railroad track appearance where there were two dark lines with a lighter band between. Usually proteins appear dark and phospholipids appear light. This led to proposing the protein sandwich model we have just mentioned. However, this model was falsified. This was because many evidences that were collected from various experiments did not fit with the model. Due to the discovery of the freeze etching technique, scientists used this technique to investigate the structure of cell membranes. In this technique, the cell membrane is split throughout the lipid bilayer. Scientists investigated the freeze-etched electron micrographs obtained and noted some bumps that exist in the middle of the layer. These bumps indicated that there were globular structures scattered at the center. This contradicts with the idea that proteins are only found on both sides. Also, when scientists extracted membrane proteins, they found that they vary in size and shape, and many had a globular shape. These globular structures were unlikely to form a continuous layer as proposed in the sandwich model. Some membrane proteins were also found to be free to move within the membrane rather than being fixed in one place. Based on all of this, the protein sandwich model proposed by Davson and Danielli was falsified and rejected. A new model was proposed by Seymour Singer and Garth Nicholson in 1972. This model was called the fluid mosaic model. In the coming video, we will go through this model in detail.